Okay, shall we get started? All right, so today the new material we're going to go over is in chapter 14, and most of the chapter, not quite the last little bit. And so we'll mostly be, the new material today will be an introduction to genetics. But again, I want to review just a little bit. I'm going to review a little bit about the DNA replication, not the whole thing, but just certain questions that have come up in office hours and things like that. So I'll start with that, and then we'll do a quick little review of part of the cell cycle, and then get going from there. One of the things, so I've gotten a couple comments about, can I post my PowerPoints the day before the lecture? The problem is the first time I'm teaching it, so it's not like I have the PowerPoints. If I had them done, I would. I usually end up getting them completely finished, not too far ahead of the lecture. But what I'm going to try to do is the night before post up a pre-version that probably will be most of the slides, so you can have that if you want to write on. And then after the lecture, I'll post the final version, which is actually what I will have used. Okay. But that means that if you're on your computer and just trying to depend on that, they won't be exactly what I'm using. But if you're trying to take notes, most of the slides should be there. So hopefully that'll be okay with you guys. All right, so back again, we talked about the idea here of a cell dividing and it going through this cell cycle, and the couple of the important things that have to happen for a cell to divide. So the first thing we actually talked about was the idea of DNA synthesis and replicating your DNA, right? And so we gave this model of this semi-conservative mechanism of DNA replication where you would break the strands apart, and there was a relatively logical way to make an actual identical copy of the other strand then. And we talked about the way that either a circular piece of DNA or a linear piece of DNA, when it's replicating, there'll be an opening in that where the strands will come apart and you'll get a replication fork and you start going around it. So one of the questions that a couple people have asked me is that if you have a small circular DNA, do you really need Okazaki fragments? Do you really have to do it where you have a leading and lagging strand? And the answer is yes. Okay. And part of the reason for that is that if you only tried to go in one direction or in leading strands around the circle, you'd end up with a large part of it being double-stranded, a large part being single-stranded until the polymerase came all the way back around. And you don't want that because you're going to leave the single strand relatively unprotected. And the other reason it's not going to happen is the primers are still going to form. That enzyme doesn't know which way the fork is going, so it's still going to start making little primers of RNA, and those are going to start to extend, and you'll get lagging strand as the fork is opening, whether it's a circular piece of DNA or a linear piece of DNA. Okay, so again, that primase is going to sit down. It doesn't know which direction the fork is going, so you're going to start getting a leading strand, but then on the other side you're going to get Okazaki fragments. So again, the way that the primase works is that it can start synthesizing RNA without there being any double-stranded region. And the reason you have to start that way is that the polymerase, which will come on later, it can't start if there's not already a double-stranded piece there somewhere. But it's fine if it's a double-stranded small piece between an RNA string and then the DNA template there, and then polymerase can get started. Okay, and then again, the primase is going to do the exact same thing on this side, even though that's the lagging strand, and you're going to start extending, and that's going to give you these Okazaki fragments. And so in something like E. coli, which is circular, those Okazaki fragments are something like 1 to 2,000 base pairs long, and in humans, the Okazaki fragments are much smaller. They're 100 to 200 nucleotides long, but in both cases, you make them. So any way that you're replicating the DNA, any inside sort of circle or chromosome you're replicating, you're going to get a leading and lagging strand. Okay, so then we talked about once you replicated the DNA, then you had to go through mitosis, and the part of the goal of mitosis was to get the DNA equally partitioned between the two cells, and not just an equal amount of DNA, but in fact, each copy had to be segregated so that one copy went to each cell. And so we talked about the phases of mitosis, and that what you did is you condensed the chromosomes together into sister chromatids. So those were the two copies of the double-stranded DNA that had been produced by the replication process, and you lined them up, and then you pulled them apart, and you did it in such a way that one copy went to each cell. And there's a little movie I'm going to play, and unfortunately the sound isn't working, so I will attempt to narrate instead. And so we can start it. Okay, so it starts off with somebody hurting their knee and getting a cut. All right, and the idea is that the reason you're able to heal is that cells can divide in response to an injury, right? And so they'll start to undergo mitosis. And so if we look in a cell, then the first step is for that cell to actually enter DNA replication. So you're going to start opening up replication forks, okay, and start making two copies of DNA. And again, remember, it's sort of loosely wound DNA. It's not organized into a condensed chromosome at this point. And so it's going to actually go ahead and replicate, and then you're going to rewind it, okay, and condense it down into chromosomes. Okay. And so then you start to get the more ordered structure. And by the time it's doing that, of course, it is already replicated. And then you get the sister chromatids, and then you enter the mitotic cycle, and so the centrosomes have replicated, and they start to migrate around the two sides of the nucleus, and the nuclear envelope starts to break down. And then these are the microtubule spindles that go between them. Okay? And again, the DNA is continuing to condense, and now starts to look like those chromosomes that you see in the micrographs and in drawings. Okay? And the sister chromatids are here, and they're held together by that cohesin protein that keeps the sisters together. Okay? And so yeah, now the nuclear envelope is breaking down, and now some of these microtubules will attach to the kinetochores of those chromosomes. Okay? And they're going to start pushing and pulling on them and getting them lined up into the middle, into the metaphase plate. Okay? And they tug back and forth. And we'll talk later that there's really a checkpoint that the cell can know that all the chromosomes have been attached. Okay? Because it'd be a mistake to start dividing if a chromosome hadn't yet been attached. Both copies, one copy would go one way, the other copy might get lost. Okay, so then they're going to line up, and you've got microtubules that don't connect to chromosomes that just go straight across, and other ones that go to the chromosomes. And then the ones that connect to the chromosomes will start to shorten. So the cohesin will disappear, and now the chromosomes will get pulled apart. All right? And they'll start heading towards the two poles. And these microtubules that aren't connected to chromosomes actually elongate, pushing the centrosomes further apart from each other. All right, and now you've got a copy of each chromosome going to each pole, and now you're going to go through the last phase where you're actually going to divide the cell. So you're going to get, in an animal cell, you'll get a constriction, and then that constriction will divide the cell into two, and the DNA will decondense, and the nuclear envelope will reform. Okay, so that's just a quick summary again of the events of mitosis. And then we talked about if you were able to look at how the microtubules are changing length, that in fact the shortening is occurring close to the chromosomes. So what's happening is that where the microtubules are connected to the kinetochore, you're actually now cleaving the tubulin off, and you have a motor protein that's pulling it. And I think I misspoke, and I said the motor protein was something like myosin, but myosin is a motor on actin, not on tubulin. And somebody kind of pointed that out to me, and it's the motors are kinesin and dynein, primarily kinesin, and that's the molecule that's then pulling the kinetochore and of course the whole chromosome then in this direction towards the centrosome, and then at the same time that the microtubule is depolymerizing at that end and then getting pulled. All right. Okay, so of course not all cells, that's fine for an animal cell. We showed that for a plant cell it's very similar, except you got a cell wall forming in between the two. But if you have something like E. coli, there's no nuclear envelope. And in fact, what happens is that the origins of replication end up attaching to the membrane at the two ends, and then as the cell undergoes cytokinesis, that's part of what pulls them apart, and you don't have that traditional kind of looking nice microtubule system in there. And so in fact, in other single-cell organisms and in various other eukaryotes, you actually find different ways of pulling them apart, and the details aren't so important. It's just the point is that there are some variations on this theme. So for example, in diatoms and some yeast, in fact, that whole microtubule assembly is inside the nuclear envelope. The nuclear envelope doesn't break down, and it actually divides as a nucleus. Okay? So you can do a variety of things like that. that. Okay, so that gets us to dividing a cell. And if you're a single cell organism, that's perfectly fine then for reproduction because you're taking one bacterial cell, for example, and making two. And the two are more or less identical to one another. What makes them different is there's occasionally errors in DNA replication, and so there's an accumulation every now and then of a mutation. And then one daughter will inherit that mutation, and that's the only way that you'll really get differences between the two daughters. Otherwise, they're usually faithful copies of each other. But as you realize that that's different for, say, animals like ourselves and multicellular animals that actually undergo sexual reproduction instead. And so what we're going to talk about today is predicting the genetic outcome of sexual reproduction. So what happens when an animal reproduces in terms of the genes that it inherits. Okay? And so you're all familiar then with the fusion of sperm and egg or pollen and the developing the egg of the plant cell and then forming an embryo. And when you look at the outcome of that, right, so mom and dad and puppies, and there's an obvious thing that the offspring look something like the parents, okay? But they don't have to look exactly like the parents, all right? And so, for example, if you made a tiger and a lion together, you get these tigons and ligers, depending on which way you go. And you can see that these animals aren't totally like one parent or the other. They're actually a mixture of the characteristics of the two, all right? So the question is, how predictable is that outcome, okay? When you have sexual reproduction, how do you know what the offspring is going to inherit? And so to start that, we start with a story that you probably all heard something about, which is the discovery of the mechanism of inheritance, or the patterns of inheritance. And that was discovered or first elucidated by Gregor Mendel, okay? So Mendel... he was a monk, but he'd gone to university and he'd studied basic aspects of biology, science, and was also well trained in physics. And the important part of that is that it meant he was actually really good at math. Okay, and that was the key, is that he actually looked for patterns in what was going on, but could actually quantitate them and then come to an understanding of what the numbers meant. All right, and that was really, I think, one of the big breakthroughs that Mendel had, was that he did the same process, but people had been breeding animals forever, and they realized some basic rules, right? But they didn't have a real formal framework to say what the outcome was really going to be. And so that was Mendel's breakthrough, is that he figured out how you could actually predict the outcome, right? In a relatively simple way, but it's an incredibly powerful tool that he came up with in terms of being able to understand what happens in crosses in reproduction. Okay, so basically what Mendel was trying to understand is principles that account for the passing of traits from parents to offspring, all right? And before him, there had been this idea that, well, somehow traits just blended, all right? So, but no real formal quantitative way to say how they were going to blend. And what Mendel did was he came up with this hypothesis that traits were really particulate, that they could be divided sort of into a quanta, into particular traits that could be followed in very specific ways, right? And this, one of the things that hypothesis does, as we'll get to, is it can account for the reappearance of traits when they're not in the parent. So a classic example would be, two parents who have dark hair have a child with red hair. Well, that's not so surprising if you realize that some of the grandparents and great-grandparents had red hair, right? Nobody's surprised by that. But why exactly did that happen? And Mendel lets us understand that. Okay, and he did this with garden peas. So it seems really sort of banal that you would be doing this with peas, but there's some very obvious reasons to do it with peas. They're really easy to grow, for example, and you'll see in a minute that their generation time is relatively rapid, and he could do some certain things with peas that would be hard with other plants or animals. Okay, in doing this, he discovered the basic principles of heredity, and the key was also carefully planning his experiments once he realized that he had certain hypotheses that he could test, and then again following the numbers, being able to do that. Okay, so the advantages for peas, right? It was realized that there are many varieties of peas, right, out there, but they have distinct heritable features that people sort of already understood, because they would raise peas and look at their traits and breed them, not knowing exactly what was going to happen, but having some idea of how to breed them. And you could get certain characters like flower color, okay, that we'll talk about. And these characters, these variants, right, like having purple flowers or white flowers, we call traits, okay. And in the peas, the mating can be controlled, and we'll talk about that in a second. And each flower actually can produce both sperm and eggs, right. So a flower has the ability to do both, all right. And so certain plants can self, meaning that the pollen can actually fertilize that same flower, and you can control that process, okay. But you can also cross-pollinate, so you can also take pollen from one flower and use it to fertilize a different flower, all right. So you can have this sort of controlled mating. All right. So we'll go through this now, how he does this. All right. So the way to control the mating, so let's say we have this purple flower, we want to mate it to this white flower, pea with a purple flower, pea with a white flower. So you have the stamens and carpels, right? And the stamens are going to make the pollen, and the carpel is where basically the egg is going to be, the pollen has to fertilize. You can avoid selfing by cutting off those carpels when they're forming, and so then that flower won't have its own pollen. And then you can take a paintbrush and take pollen from this other plant and spread it onto here, and then you know that in fact all the fertilization has been cross-fertilization. Okay? So you can control that process. Likewise, you could actually take this flower, close it up in a little bag, and then the only way it could get fertilized is with itself. So then you could self-pollinate. You can do a cross that way. So you can do both kinds of things. Okay, so when you do a cross, then you call this first generation F1, or first filial generation. And so the idea is, well, what's going to happen if I cross a white flower and a purple flower plant? What am I going to get? All right. But in doing this, Mendel chose to track traits that only had two distinct alternative forms. All right. So that was important to simplify the problem. So it would be more complicated if there were like 5 different colors of flowers and you were trying to do the cross. Right. So now we could understand how to do that. But in discovering this process, Mendel was careful to pick traits that just had 2 states to them. Okay. And then he also started, and this was very important, he started with varieties that were true breeding. Okay. And so what does that mean? That means that he started with, say, purple pea plants, that when they were self to each other, they only gave rise to purple flower pea plants. Okay. And he started with another batch of peas that had white flowers, and when they fertilized themselves, they only gave rise to white flowers. Okay. So that's called true breeding. All right. And so he started with those, and he did crosses. Okay. And so what he would do typically is he would make 2 contrasting true breeding varieties. Okay. And people call this process hybridization. All right. So the idea was that you would start with a true breeding purple flower plant and a true breeding white flower plant, and he would now make them to one another and ask what would happen now. Okay. And so we call the parents the P generation, and then the first generation hybrid is the F1. Okay. And then if we cross those F1s to each other, we call those the F2. Okay. The second filial generation. All right. So what we're going to do is follow crosses in F1 and F2 and see if we can understand what happens. Okay. So in just one example, okay, he would take a true breeding white and purple flower pea plants, and what he noticed was all the F1s were purple. Okay. So he had a true breeding white, true breeding purple, across those, and those were all purple. Okay. But then when he mated those F1s to each other and got an F2 generation, what he found is that he had both purple and white flowers now appearing. Okay. But he noticed that there was a very set ratio of 3 to 1. Okay. So in the F2 generation, he found that there was, for every 3 purple flowers, there'd be 1 white flower in all those plants. Right. And he had reasonably large numbers, and they were close to that kind of ratio. So again, this is the sort of drawn out cross. So you take this true breeding purple, true breeding white, and cross them to one another, and now you notice that in the F1 they're all purple. So they all look like one parent and not the other. All right. And in fact, you couldn't tell these apart, all right, from each other. But Now, and remember, if you were just to mate these to themselves, right, the true breeding population, they'd always all be purple. But if he takes these guys, the F1s, and mates them to one another, now what he sees is he gets a ratio of 3 to 1 of purple to white. Okay, and so that's what he observed. So Mendel reasoned from that that only the purple flower factor was affecting flower color in the F1 hybrids, meaning that there was a single trait that was being segregated. Okay, and he came up with this idea of dominant and recessive. So he said that the purple color is dominant to the white color. Okay, or the purple trait is dominant to the white trait. Okay, but that as you go through generations, the factor, the way he put it, for white wasn't destroyed. Right, it could reappear in the next generation. So it was hidden somehow, but then it could reappear in the F2. Okay, and in the end, what Mendel did is what he published was 7 different traits. So the purple flower and 6 other traits. And what he found was a fairly consistent set of rules for those 7 traits. And we'll get back to that Mendel was actually a bit lucky, but there was he clearly had the insight in looking at his data to understand what was going on. All right, and what Mendel called heritable factors, we now call genes. Okay, so these were the loci that he was following. So he just called them heritable factors. We now generally think of them as genes that we're seeing the segregation of. Okay, and these were the actual traits that he had. It's not so important, all of them. But so there was the flower color, there was flower position, seed color, seed shape, pod shape, and pod color, green versus yellow, and stem length, long versus short. And those were the factors that he followed, the 7 different ones. And he came up with sort of a set of rules. So he developed a hypothesis to explain this 3 to 1 inheritance pattern that you saw in the F2. Okay, and he came up with concepts about how that model worked. And this is now related to our understanding of genes and chromosomes, which we're going to go through. Okay, so first he said that there are alternate versions of genes account for various, or now we understand it as genes, alternate versions of genes account for variations in inherited color. So for example, the flower color in a pea exists in 2 versions, right? One for purple flowers and one for white flowers. But it's the same trait. It has 2 versions of it, a purple version and a white version, right? And so we can think of this as the gene that's controlling this. And there's one, what we call allele, that gives us purple, and another allele that gives us white, okay? So again, the alternate versions of these genes are now called alleles. And then each gene resides in a specific place on a specific chromosome. And that's what we understand now. Mendel didn't know that, but that's what we now understand. Okay, so we can look at it this way. And again, remember that this is a fiction of the DNA in a cell, other than mitosis is condensed into this chromosome structure. But if you look at it, if it were, this is what it would look like. And you have 2 copies of the same chromosome per cell. Okay, but if you looked at an individual gene on that chromosome, okay, so maybe most of the time they'd be the same, but sometimes you have allele differences. So in this case, you have an allele for purple flowers on one chromosome, and you have an allele for white flowers on the other chromosome. Okay, so those are the 2 traits. And this would be an animal that had both alleles in it. Okay, a true breeding purple flower, the chromosomes would only have this purple allele, and a true breeding white flower, all the cells would only have that white allele. Okay, all right. And then he said that for each character, an organism inherits 2 alleles, one from each parent. Okay, and so this was an important insight that came from the numbers, that an organism actually had 2 alleles for each locus, and one allele came from one parent, and the other allele came from the other parent. Okay, and he figured this out without actually knowing anything about chromosomes or what was inside a cell or anything like that. Right? This was purely sort of analyzing the data of the phenotypes that he got. Okay, and then 2 alleles of a particular locus may be identical in the true breeding, right? So an animal that only made purple offspring, the 2 alleles were both the purple form, and an animal, a pea plant that then when it was crossed to itself and you got more and more progeny, they were all white. So then the alleles that it had were only the white alleles. But alternatively, like in the hybrids that you got between a purple and a white flower, then you would have an F1 that was a hybrid that had both the purple allele and the white allele in it. Okay, but if you had these 2 different alleles in an animal or plant, right, that one would be dominant to the other. So if you had a purple and a white, the flower would actually be purple because purple was dominant to white. Okay, and the reason is again that so by definition that allele is the dominant allele then. Okay, and then the 4th thing that he realized was what we call the law of segregation. So the 2 alleles for a heritable character segregate during the formation of gametes. So those are the sperm and the egg. Okay, so you have 2 alleles in the animal, but then when you make a gamete, you separate those 2 alleles, and an individual sperm or individual egg only gets one of those 2 alleles, not both. Okay, and this segregation actually corresponds to something that we're going to actually talk about a little later in the course, which is meiosis, which is we'll talk about how chromosomes actually do this, how you go from having 2 copies in a cell to making gametes that only have 1 copy. Right? And that's going to be called meiosis, and we'll go over that later. Okay, so his segregation model accounted for this 3 to 1 ratio that he observed in the F2. Okay, and what we'll do now is go through a way of looking at this sort of graphically and of calculating the probabilities of what's going to happen. This is a Punnett square. And again, those of you taking the lab have already gone through this at some level. And so we'll go through this here, and then we'll get more complicated examples as we go along. And the way the book does it, which I'll follow for today, right, which is I think a little different than the way you guys do it in the lab, is that we use a capital letter for the dominant allele, and we use a lowercase letter for the recessive allele. All right? So then we can write it like this. So if we have a true breeding purple flower, then it's capital P, capital P. Okay? So it has 2 copies of the allele, and they're both capital P. And a white flower we would designate as the recessive allele, and it's lowercase p, lowercase p. Again, 2 copies, right? And so that explains why if we make these together, they're all purple. If we make these together, they're all white, because those are the only alleles you have. But if you make the cross, right, then what's going to happen is that all of the gametes from this animal are going to be big P, and all the gametes from this animal are going to be little p. And that doesn't matter whether they're the sperm or the egg, or the pollen or the egg, okay, in this case. And so you make them, and then you get all purple flowers. Okay? And the reason that they're all purple is because all of these plants are big P, little p. Okay? So they have 1 copy of each. And so why is it purple, though, instead of white? Okay, right.